For decades, there's been this huge question lurking in the world of genetics, a real mystery hidden right on the surface of our DNA. And solving it, well, that could change everything about how we treat disease.
So the whole debate boiled down to these tiny little chemical tags, they're called methyl groups, that latch onto our genes. For years, scientists were split. Were these tags just, harmless gunk that builds up on genes that are already switched off? Or were they something way more powerful, the very things holding those genes down, locking them in the off position?
So you had these two camps. On one side, you had the cobwebs theory. The idea was that these methyl groups are just a side effect. They show up after a gene goes silent. But the anchors theory, that suggested they were the cause, that they were actively holding the gene down, keeping it turned off. And figuring this out wasn't just for bragging rights. It had massive implications for how we might treat genetic diseases. To finally get an answer, researchers needed a completely new kind of tool, something a lot more delicate. Think less like a hammer and more like a feather duster. And that tool? It's a third generation of CRISPR technology, and it is an absolute game changer. It's actually pretty cool to see how this technology has gotten more and more precise over time. The first version of CRISPR, that was like a pair of molecular scissors, literally cutting broken bits of DNA. The second generation was more like a pencil with an eraser, able to fix single letters in the code. But this third generation, this is what we call epigenetic editing. It's totally different. It works on the chemistry around the DNA without ever touching the code itself. And that difference is just huge. Traditional CRISPR literally snips the DNA strand. It's powerful, yeah, but it's also risky. Cutting DNA can lead to all sorts of unintended mistakes and other health problems down the line. This new method is more like a genetic eraser. It doesn't cut a single thing, it just wipes away those chemical tags, leaving the DNA sequence perfectly intact. So armed with this incredibly precise new tool, researchers from UNSW Sydney and St. Jude Children's Research Hospital could finally put that whole cobwebs versus anchors debate to the ultimate test. And the experiment they came up with was just beautiful in its simplicity. So first, they took a gene that was totally silent and covered in those methyl tags. Then, using their new epigenetic editor, they just erased the tags, and boom, the gene switched right back on. But here's the real kicker. They then used another tool to put the tags back on, and just like clockwork, the gene switched off again. The lead researcher, Professor Merlin Crossley, put it perfectly. He said the mystery was solved. These tags weren't just passive cobwebs at all. They're molecular anchors, actively pinning the gene down in the off position. All you have to do is brush them away, and the gene just springs back to life. Okay, so this wasn't just some abstract scientific discovery. It immediately blew the doors open for a whole new way to think about therapies, especially for a really devastating genetic condition, sickle cell disease. See, sickle cell disease is caused by a faulty gene that messes up your adult hemoglobin. This makes red blood cells get all hard and crescent-shaped, and they can't carry oxygen properly. It leads to blockages, unbelievable pain, and a whole list of life-threatening problems. But what if you could just tell the body to use a different, healthy gene instead? Professor Crossley has this fantastic way of explaining it. We all have a gene for fetal hemoglobin, which works perfectly when we're babies. But soon after we're born, it gets shut down by those methyl anchors. For someone with sickle cell, that sleeping gene is like a perfectly good set of training wheels that's just been tucked away in the garage. This new tech gives us a way to get them back on the bike. So here's how a future therapy could work. Doctors would take some of a patient's own blood stem cells. Then, in the lab, they'd use this genetic eraser to wipe the anchors off that sleeping fetal globin gene. With that healthy gene now switched back on, they return the edited cells to the patient, and those cells can then start making healthy, normal red blood cells. No cutting, no risk of permanent DNA damage, just waking up a healthy gene that was there the whole time. The ripple effects here go so far beyond just one disease. We are really looking at a fundamental shift in how we even think about gene therapy. So let's just recap why this is such a big deal. First, it finally settles a huge decades-long debate in genetics. Second, it gives us a much safer way to do gene therapy because we're not cutting DNA anymore. It creates a real path forward for treating sickle cell, and maybe most exciting of all, it opens the door to treating countless other genetic diseases that are caused by perfectly good genes just being silenced. As Professor Crossley himself said, this isn't just another small step. This is the very beginning of a new age, a leap into a whole new world of possibilities for medicine. And that leaves us with a pretty incredible thought, right? For the very first time, we have a precise, safe, and even reversible way to control our genes. We've gone from trying to cut the wires to just learning how to flip the switches. And the question now is, what other doors is this new key about to unlock?